I think I mentioned last week how they were looking at the microbes hanging out in termite guts. And so there's been lots of insects that have been studied. Aphids are one of them. And believe it or not, this one insect is the cause of a lot of headache and crop loss every single year. So they're tiny little insects. You can see them here, different stages of their development, some larvae, as well as the adult stage and some younger stages. And so they're difficult to see. Those of you who work in the greenhouse and have been in the greenhouse, I'm sure you know what I'm talking about, but you might have to, if you've never looked for them before, generally looking on the underside of the leaf is a good spot for them. And they like to put their needle-like mouthpiece into the vascular system, into the veins, and start sucking away all the nutrients. Primarily, they're going for the sugar. And so they're a big problem. One of the issues is that they gain resistance to pesticides very quickly, especially the ones that hang out in greenhouses. They're well known for that ability. So it is a major pest. And the phloem vessels, that's part of the vein. That's the phloem is where you see the sugar coursing through the body of the plant. And so that's a very attractive nutrient source for the aphid. And what they found is that the aphid has this kind of glandular tissue near their mouth part. That's called the mycetome. And I don't know quite why they call it that, because it makes you think about fungi instead of bacteria, because MYC, usually you start thinking about something fungus-like with that spelling in science. But anyways, if you cut that open and look in, there's bacteria hanging out in there. And what they found is it's primarily this bacteria, Buchnera aphidicola. And so they even named it, after its host species. And Buchnera is necessary for the aphid because even though it's pretty to think that we can all live just on sugar, including aphids, aphids can't just live on sugar. So as they're taking the sugar out of the phloem, that's not a well-rounded diet for the aphid. They need some other things. And so what they started doing is they're like, well, how important is this bacteria? And so they would do experiments where they treat the aphid with antibiotics and to kill the bacteria. And if they did that, the aphid would die. And so then they started going, okay, clearly this bacteria seems to be pretty necessary. And so they started looking at what is the bacteria providing to the aphid in order for the aphid to survive. And they found out that there's key amino acids and growth factors that the bacteria are making and excreting out, and the aphid can take that up and use it. And so without them, they don't make it. So my question is, why don't they take advantage of this? You'd think there'd be an insecticide that would target, rather than the aphid, the actual bacteria. But I have not seen anything on the market that does that. And I wonder if this knowledge is just not out there that much. But it seems to me that would be another method of attack. Instead of trying to kill the aphid, kill the bacteria in the aphid, and that would kill the aphid. But I have yet to see any kind of product advertised that way. So I don't know. Sounds like a niche that needs to be, there could be some profit there if someone could come up with a strategy, because this is a big deal. It's not just with greenhouses, it's crop fields. Aphids can really be disruptive to your growing season for sure. This is not an aphid. This is coming from, I think this is a leech mouth part, but I just wanted to show the idea of this kind of glandular-like tissue. That's called the mycetome. And so it's near the mouth part. And if you look inside, all of this kind of long different rod shaped, that's all there in there. You can imagine that in the mycetome tissue. So, it's not just termites that need their bacteria in order to survive. Here's another example of that. So very tight relationship. The aphid host definitely needs its microbial partner. We talked a little bit about this idea already, where you have protozoa that can have endosymbionts. We looked at a protist with the algae inside and how the algae can do photosynthesis, and it's really good because it can provide oxygen. And here's an example of protozoa with bacteria inside, and the bacteria are kind of that reddish orange color. And they're finding the more they look, it seems to be a pretty common strategy. And a lot of evolutionary biologists are studying this because they think that maybe this could have something to do with the evolution of organelles inside the eukaryotic cell type, having this. And, we've already learned about that with Lynn Margulis, Dr. Lynn Margulis, and her endosymbiosis theory of how the mitochondria and chloroplast arose. And so they're studying this carefully to kind of support that idea and see there's other ideas out there that the nucleus could have been a free living microbe at one point, flagella also. And so there's other ideas out there that they're pursuing to see if they can find data to support some of these hypotheses. And certainly for medical applications, it's a nice system to study. It's relatively easy to study in a lab environment. They know how to grow them and work with them. So it's really kind of a nice research tool for them to try to get at some pretty important questions. So the actual organisms that are being shown here, you don't need to know the names of them, but it's a acanthamoeba protist, and then the bacteria is called Protochlamydia. Okay. So we have other examples of microbes that hang out in more sophisticated eukaryotic organisms like corals and hydras and anemones. So any of you who've had the good fortune to scuba or snorkel with coral reefs, you know that it can be an incredibly beautiful area. It's an incredibly important area for ecosystems in marine environments. And what they find is that it's pretty universal that there will be endosymbiotic algae within these animals that form the coral reef. Sponges have been studied. They know that there's cyanobacteria inside. And I would argue that probably the corals are the best studied, primarily because of coral bleaching issues. The research on corals has really skyrocketed. And here they know that there's certain type of algae called dinoflagellates. They're a special group of algae that hang out in the coral animal on the coral reef. And they're algae, so the dinoflagellates, they're capable of doing photosynthesis. And so all the sugars that they get from that, they can provide. They release oxygen, so that's good too, because as we know, oxygen is slow to diffuse in water. So having that right there inside the coral is a great boon of a good source of oxygen. They can help remove extra ammonia. Ammonia is toxic to any organism in the right dose. And so they keep that down to good levels. And they tend to call the tissue that they live in the symbiosome. That's kind of a common term that you see in the literature. And so this interaction, the corals and the algae, have been very intensely studied. The coral, as the host, is providing physical protection, and they help get access to sunlight because as they extend their polyps out of the tougher coral body during the day, they'll extend their polyps, and then that will help the dinoflagellate do its photosynthesis, gets good access to sunlight. So phototaxis is just the idea that the polyps are movable and they can move toward the best light that they have around them. And they definitely see correlation with if the algae are growing well and seem really healthy, the corals seem healthy and grow in a similar way. And so this interaction really started to get more heavily studied when they started looking at coral bleaching So these are healthy corals. These are pictures of healthy corals. They're not, the dinoflagellates don't necessarily always look green, although the top picture, you can see that. But sometimes they have other pigments that obscure the chlorophyll that's present in the algae. And so sometimes they look more brownish, but that's not a damaged coral. That's just fine. And you can see how the polyps are extended, trying to get access to sunlight. And so the phenomenon of coral bleaching has been sadly going on for a while now, and it left scientists scrambling to figure out what would be causing that. Seems like the kind of the main idea here is that it's not one thing, it's a mixture of unfortunate events. So changing temperature, where temperature goes up, you have runoff and pollution. Too much sunlight can be bad for even organisms that need to do photosynthesis. That can be a problem. And even they're looking at the idea of extreme low tides in areas where there's coral reefs and in more shallow areas that they're not fully covered by the ocean water anymore. And so it's a mixture of things. And here they're showing the kind of the gradual decline over time, and they are calling it bleaching because essentially what ends up happening is the dinoflagellates, the algae inside, die off and they just kind of disappear. And so then the coral is left without their partner. Not a good situation. And so this is something that they've been chasing down and trying to figure out, not only what causes coral bleaching, but then, what can be done in order to try to support the coral and have them come back from being stressed and bleached. And can we do that? And so some scientists had the great idea to start thinking about the microbiome of corals, not just the algae. Certainly that's part of the microbiome of the coral, but all the other microbes that hang out as well. So it's not just algae. But early research, they just kind of ignored all of those organisms and just looked at the coral and the dinoflagellate. And then they started looking, some groups of scientists said, well, what about the other microbes that are hanging out with the coral, both on the surface of the coral and in the tissue of the coral? And so here is a paper that was published. This one was 2020, where they're saying, hey, maybe we should look at this idea of a disturbance in the microbiome of the coral. So that fancy word dysbiosis just means that it's a disturbance and an imbalance of the normal microbes that hang out with the coral in this case. And the holobiont idea, I'll talk more about that when we get into microbe-human interactions. I'll start defining that a little bit more. But I was really happy to see that in the title of a science, published paper, because this says the holobiont idea has been proposed relatively recently and initially met with some pushback, but now it's becoming more accepted that when you think about any organism, whether it's a plant or an animal of some sort, including humans, you have to think about the associated normal microbes that hang out long-term with that host. And think of that whole organism as a superorganism and as a unit. And that's the kind of the idea of the holobiont. So you have to think about the entire organism with all of its microbiome. And so here they're looking at the idea of dysbiosis, and that as the coral is getting stressed by these environmental stresses, that the microbes, in addition to the algae, are shifting. And that if we can better understand that, then perhaps we can find ways to help reintroduce the right partners back to the coral, kind of like a probiotic idea. And they're looking at this for trying to build coral reefs back. There's people who grow coral reefs and then they go and outplant them, very much like, planting crops in a field. They're doing that kind of work to try to save the coral reefs and reintroduce, healthy coral. So this is that paper that I had in the readings. Here's one of the images. So on the upper left, figure A, that's healthy. So even though it's brown, that's healthy. That's just the color of the algae. And then you can see the bleaching going on in figure B. And, obviously you can see why they call it bleaching. Everything kind of goes white. And then C, D, and E are, C is healthy. The polyps extending and the algae are in there, everything's good. And then D and E are progressively worse bleaching processes going on. So coral reef ecosystems are hugely important in marine ecosystems. They provide so much diversity and so many services to the organisms living in the oceans that there's, rightfully so, huge concern about all of the big bleaching events that are going on across the world in these different interactions. They're even looking at quorum sensing and quorum quenching and looking at this idea going on within the coral microbes. And they're asking the question, can we use this information to help restore the proper microbial community to the algae? And so I didn't talk too much about quorum quenching. We did learn a little bit about quorum sensing, that's their way of communicating. And, the kind of the classic example of that is pathogens in you, like strep throat. They're doing quorum sensing with each other to figure out how many of their population is in your body before they mount an attack, because our sophisticated immune system could wipe out a low enough population. If they get to a high enough population, then they can all start kicking in their virulence factors, start attacking your tissues and cells, and be more successful. So it's the same idea here. They're looking at, the pathogens that might be attacking coral. So you can imagine if corals start to get sick, they do have an immune system. It's not nearly as sophisticated as ours. They don't really have adaptive immunity like we do, but they do have an immune system. But, they can reach the tipping point where they get just so sick that they're not going to survive. And then coral bleaching can really wipe them out because they're already weakened. And so the idea here is, if we can understand this a little bit better, the idea of quorum quenching is to stop the pathogens from being able to talk to each other. If you can mess with their communication, just like in warfare, if you mess with the enemy's communication so they don't know where their other units of, where the fighting is going on, then it's, hard for them to mount an attack, a coordinated attack. And so quorum quenching ideas, they're looking at that. Can we stop the pathogens? Can we inoculate the coral with beneficial microbes, kind of like a probiotic idea? And then can we help the coral survive illness, pathogen attack, and coral bleaching? So they're looking at, not just the idea of all the abiotic stresses like heat, pollution. Certainly that's playing a role, but they're saying that once the coral gets weakened, then the pathogens can really ramp up and gain a foothold. And so let's understand this better. So I thought that was a great thing to see. I was really excited that they're looking at that angle. And then this idea, the probiotic idea, how can we understand who's there? What are they doing? How are they helping the coral? And then can we find like a cocktail mixture of beneficial microbes in order to inoculate the algae and have them support the algae and hopefully reduce the amount of coral bleaching that's going on. Of course, we have to do, reduce climate change and pollution and all of that, but it's a multi-faceted approach. You can't just do one thing and hope it's going to work. There's a lot involved in this whole idea. So beneficial microbes for corals, they've even gave it an acronym, BMCs. So probiotics for corals, that's what they're getting at. I think, that's a great idea. That something that has been overlooked, but now that's starting to gain a foothold in the scientific community. So interesting work. Hopefully we'll see some good results soon. Here's another ocean dwelling organism. you'd have to sign up for a submarine trip for this. This is deep, deep down in near the hydrothermal vents in the sea floor, ocean floor. And we're looking at tube worms that are collecting near these hydrothermal vents. And it's not just tube worms that live here. So when they first came, you know, if you've ever seen video of submarines going along the ocean floor, it's just like darkness, like the surface of the moon. There's like nothing there. And then all of a sudden they come up to a hydrothermal vent and there's just this massive activity of all kinds of animals and organisms that are hanging out there. And so that hydrothermal vent, that water that's coming from the ocean, I mean, the Earth's crust up through the vent is very nutritious from a chemolithotrophic perspective. So there's a lot of minerals that are coming up. And we know that there's bacteria that can take those minerals and strip electrons off of them and put it into an electron transport chain process, and they get their energy. And so as that very hot water is coming up, you have dissolved minerals in there. And so things like hydrogen sulfide gas, hydrogen gas, ammonia or ammonium. So these are reduced minerals. That means there's electrons there that can be stripped off. You can take electrons off of reduced molecules and feed them into an electron transport chain. So when they started looking at these animals like the tube worms, they found that they do have chemolithotrophic bacteria hanging out in specialized tissue of the tube worm. And the bacteria are busy stripping electrons off of hydrogen sulfide, which is toxic, by the way. So if there's too much of that around the animals, they'll die. So it's a good thing the bacteria can use that and break it down. And so there's a couple of genera, Beggiatoa and Thiobacillus, are found with tube worms. And so they've been studying those quite heavily. But it's not just tube worms. There's all kinds of clams and mussels and other organisms too. And it's amazing if you ever look on YouTube for those types of videos. It's really quite stunning to see this little oasis in the ocean floor. I think I showed this picture already, probably during week 1. And so here, just reminding you that the ocean floor where you have these hydrothermal vents, it depends on what kinds of minerals are coming up with the hot water and how hot it gets, what you'll look at. You can have so-called warm vents that aren't quite as hot. And then the really hot ones are typically called black smokers because they actually have plumes of black gases and liquids that come up that look very distinct. You can clearly see that. And you can see here they're showing iron sulfide, hydrogen sulfide. And so those dissolved minerals are in their reduced forms and can be mined for electrons, so to speak. And so that's a picture of a black smoker, very prolific. And then here on the right picture, all the green are bacteria, and then all the orange are microbes in the domain Archaea. As we learned, Archaea tend to dominate in the lower levels of the ocean. So they tend to find quite a bit of those present. So when they first saw all these organisms living that deep and then living in areas, you know, where there's not a lot of nutrients, and then they started investigating the vents more, and then they're like, oh, well, in this region of the ocean floor, there are a fair bit of nutrients, but you would need the chemolithotrophs to act on them. And so they started taking samples of these animals and looking for microbes in the tissues. And another thing to note is the reddish plume that you see as part of their body. I'll talk a little bit more about that too. So when you look at that part and dissect it, there is a section of tissue that is called the trophosome. And when they take that and dissect it and look at it under the microscope, they found chemolithotrophic bacteria, and they're packed in there, and they extract electrons off of those dissolved minerals and run an electron transport chain. So they're oxidizing those minerals. and they can get their ATP that way. It seems that these trophosomes have like dedicated bacteria, like certain bacteria hang out with certain animals, but not with others. It seems to be that way, at least what they're studying so far. And when they look in the trophosome tissue, there's a lot of bacteria per gram of tissue, 10 to the 9th levels of bacteria cells per gram of tissue. So they're jam-packed in that trophosome tissue. And so they're trying to show how, whoops, I can't do that. Let me take that off. So here's inside the trophosome, they're trying to show how there's hydrogen sulfide compounds. And then they use oxygen at the end of an electron transport chain, because we know oxygen is the best electron acceptor. So they can get access to oxygen, ship electrons off of reduced minerals, and then they can run the different electron transport chain processes. And then you also see CO2. So they're able to take in CO2 and with an input of ATP and other molecules, they can make their sugars from that. And so they can essentially feed the worm sugars as well, the carbohydrates. So they were wondering, how does the bacteria get access to enough oxygen in order to run these electron transport chains so efficiently? And so they started looking at the tube worm, especially when they saw that reddish tissue that generally makes you often think about like blood and hemoglobin and things like that. And sure enough, they have special hemoglobin that not only binds oxygen and CO2, but also hydrogen sulfide. So we definitely, our hemoglobin is designed for oxygen delivery and CO2, to get rid of the CO2 and respire it out. But they also will have the hemoglobin bind to hydrogen sulfide. So all of that can get delivered to the bacteria. And again, if bacteria have access to hydrogen sulfide and then they have oxygen sitting at the end of their electron transport chain, they get a lot of free energy from that electron transport chain process. And so almost negative 800 free energy, and you only need negative 32 to produce an ATP. So you can see they're getting a lot of ATP out of that. And so it's a pretty nice system. Yes.
So it's actually really cool because it explains this project that I've done for special topics class last year, where basically these benthic worms are some of the only organisms that have been found to contain halogenase enzymes, which can cleave like fluorine carbon bonds, which are crazy strong bonds.
Yeah.
And they're some of the only organisms that can like leave these like bonds in nature. And it's probably because the bacteria can like it requires a lot of like electron energy, and so it's hard because the bacteria can give it so much.
Yeah, that is cool. Yeah, I mean, that's a whopping amount of free energy. And so that I could see where that could really help out that process if it's that tough to break those bonds. So yeah, it's when they look at this interaction, certainly the tube worm is also providing protection, but the fact that it has this really unique hemoglobin is certainly a benefit to the bacteria as well. And then the CO2 that the hemoglobin can also deliver is the bacteria can take that and they'll run the essentially the Calvin cycle, which I talk about in my botany classes. So those of you who've had those classes, you know a little bit about that. And so it's, their way of taking in CO2 and reducing it to carbohydrate. And that takes energy too. And so some of that free energy is used for that. But then that's the way for the animal to, the tube worm to get access to carbohydrate. Whereas, if you look at their environment, they don't really have a lot of access to that in that ocean floor environment. So really a great interaction, allowing these organisms to develop a habitat there where they can actually thrive and not just eke along. If you've seen some of the videos, these tube worm, I mean, it can be pretty massive population of these two worms all by that hydrothermal vent. Looks really cool. So this, I'm not asking you to memorize this. I'm just putting it into a visual. This is, again, just emphasizing they're extracting electrons from things like hydrogen sulfide and sending it into an electron transport chain. So that's what's being trying to be shown here with oxygen sitting at the end. And then they're trying to show that they take the CO2 and run the Kelvin cycle. So I'm not asking details on this, but I would like you to appreciate, again, remind yourself what chemolithotrophic means, what they're doing, how they're getting their ATP in the big picture idea. Okay, so lots and lots of interesting things yet to discover in the ocean systems. It's, as you know, the ocean, covers more area on Earth than terrestrial. And there's a lot of mystery and exciting things to discover yet in the oceans. So, but let's go back to land. Here's another topic that's been heavily studied for quite some time. And this is, so we're talking about animals that have slightly different gastrointestinal tract design compared to humans. And so when we talk about ruminants, cows usually come first to mind, but there's others. Sheep, goats, deer, moose, caribou, antelope, giraffe. And so these are animals that are primarily eating cellulose, which is a rather tough material. If that's their main diet, that's a lot of tough material to digest. And then you look at their enzymes, they don't really have the cellulase enzyme. And so they're kind of stuck that they eat cellulose, but they themselves can't break it down. They rely on organisms in their rumen to do that. So they have an extra section to their intestinal tract called the rumen. It's essentially it's big. It's a big anaerobic fermenting sac. And it allows them to have a partnership with really high populations of microbes that have cellulase and all these other funky enzymes that can break down all the grass and leaves and things that they eat. And so this has been a system that has been studied pretty heavily to look at this interaction between microbes and their host. Some of the microbes do get eaten by the ruminant, so the cow, for example, will eat a portion of the microbes. And so that's also kind of specialized. We can't eat high doses of microbes like in big populations primarily because we don't have the physiology to break down that much DNA that we ingest from the microbes. So this is actually what they do. They perform surgery on cows and cut into the side of the cow to get to the rumen. And then they form like this cap seal on it that you can take the cap off and then you can reach in with sterile, gloves and take samples. And they keep the cow happy. There's one like this at Michigan State University. Dan is sampling there. So he's one of the former grad students in biology, and he was at MSU. And they let him don those sterile gloves and go in and take some samples. And, if you do it correctly, it's all sterile. And again, they cap it when you've taken your sample, and the cow is well cared for. It looks like a dairy cow, I see. And so these are like long-term experiments that they do with these animals. And I thought that was a kind of a interesting idea, a way of doing this in a more humane way rather than waiting for them to die or killing them and then opening them up. So here you can see the rumen is quite big as part of the intestinal tract. And then the omasum, abomasum, those areas can help with the breakdown of some of the microbes that they'll end up eating. And then you can see too that the they show the dashed line of the cud, and so they will kind of regurgitate and re-chew their food. And then swallow it again. And so they have an interesting way of dealing with their food and having the help of all these microbes. So if you look in the rumen, again, really big populations, 1 times 10 to the 10th to 1 times 10 to the 11th per gram, bacteria per gram of fluid. And so it's a nice cozy home. It's relatively stable. And temperature, the pH is pretty good, pretty stable. It's that's pretty good pH range for lots and lots of microbes to hang out and be happy. And so here you can see the reaction pathway. They're trying to show here, if you think way back to when we were covering some physiology, glucose 6-phosphate, fructose 6-phosphate, pyruvate, that's fermentation. That's glycolysis that they're hinting at there, that they're going through glycolysis. You get pyruvate. And then once you get pyruvate, you can run all kinds of different further fermentation reactions. So that's what they're showing there with the multiple arrows, that depending on the microbe, they'll do slightly different fermentation pathways, and you can end up with all kinds of products, some that are very useful and some that aren't. So methane. So you've probably heard that cows and cattle produce a lot of the methane gas. And that's kind of true. It's not really them doing it, it's the microbes in them, but that usually doesn't get phrased that way. So, but really it's the microbes. And what you can see here, acetate, butyrate, propionate, those are fatty acids, volatile fatty acids. That's very nutritious for the cow. And so the cow will absorb some of that into its bloodstream, and that provides very, very important nutrition for the host. And so this is a fancier way of looking at it, where you can see they're saying, okay, yep, feed the cow some grass or hay, and you have all kinds of complex carbs in that. And you have to get it broken down to sugars. So the bacteria have to get it broken down. And then you can see the VFAs, volatile fatty acids, that get made. And then they're showing how it can get absorbed. And typically it gets used for protein synthesis. So here, some of these others, it says CO2 and CH4 removed by eructation to the atmosphere. Anyone know what eructation means? I had to look that one up. I'm like, what? So it means to belch or burp. And I'm like, oh, okay, that's a fancy word. Why did they just say belch, burp? There's nothing wrong with it. But anyways, so apparently, they tend to say that it's more of coming out the other end of the cow, more like a fart than a burp. But actually, a lot of it's actually through the mouth that the methane gets released through burping as they're chewing their cud and releasing some of that CO2, and the methane. So I'm sure some of it does go out that back end too, but a lot of it comes out from burping. And so they really do have some, it's hard to estimate how much cattle and cows contribute. I was trying to find some good data out there, and this is one that I found: 14.5% of all greenhouse gas emissions. And so there's been a lot of thought put into, maybe we just shouldn't have so much cattle on the planet Earth, and maybe we should not be using them as such a main staple of food, and I guess beverage too with the milk. But did you know we're the only mammal that still drinks milk after being weaned? We still choose to drink milk. Any other animal on the planet doesn't do that. I just find that really interesting. But anyways, that's another philosophical topic that we don't have time for. But something that has been pointed to for reducing greenhouse gases is just reducing them. But again, it's not really them per se, it's the microbes inside of them. And so they're doing all kinds of things. Here is just a select list of some of the microbes there. The B means bacteria domain, And then you can see there's archaea also showing up, some of the methanogens especially. But you get, a range of different things going on, different fermentation products. So going back to that, I would like you to know these three: acetate, propionate, butyrate. Okay, I do want you to know those are the three main ones being produced. And that's good because the host gets that into their bloodstream and they can use that for protein synthesis. I would like you to know those. And that's going to come up again when we look at human microbe interactions. Those same fatty acids feed us as well. And so you can see just some ideas here of different types. They kind of group them by how they run their fermentation and what they attack. So cellulose or starch, for example, succinate, methanogens. So there's also ones that can break down hemicellulose, which is related to cellulose. It's a branched polymer. All kinds of sugar fermenters are in there. Fatty acid, bacteria like to feed on fatty acids, bacteria that can break down proteins and lipids. So it's just this huge, huge consortium inside that rumen, incredibly high populations, lots of diversity. And acetate and propionate, and then the butyrate. Don't forget about butyrate, but the highest dose is acetate. You see more of that showing up than propionate or butyrate. And then some of the microbes are also producing amino acids and vitamins that the host needs. There's even some that are in there that can fix nitrogen. And so that was a surprise. They didn't expect to see that. But there are some, they're not like the main bacteria in there, but they're there. And so they're taking dinitrogen and converting that to ammonia, which then gets put into amino acids. So some of the calculations, they got excited. They're like, oh, well, this is, great. Because then if they could provide a ton of nitrogen to the host, that's great because cows need nitrogen too. But when they did the calculations, it's 10 milligrams of nitrogen per head of cattle per day. That's really small. 10 milligrams is small. So they're like, no, that's too low to be a significant source of nitrogen. And then all the ammonia that can build up over a variety of different reactions going on in the rumen, that sometimes there's a buildup of ammonia, and that can actually slow down nitrogen fixation. So they realized that they got excited, but then realized, they're not really doing a whole lot of nitrogen production for the actual host. So microbes certainly, if they fix the nitrogen and get ammonia or get access to ammonia another way, microbes are using that too. It's not just the host. And so microbes can assimilate the ammonia, get it into amino acids. And then remember that the cattle does eat a subpopulation of the microbes as they're chewing their cud. They are getting access to some of those microbes. There's even eukaryotic organisms in there. There's protozoa hanging out, which if you're a protozoa hanging out in the rumen, you have a rich source of food to prey on because they like to eat bacteria. And they tend to be obligate anaerobes because that is a pretty anaerobic environment. It's difficult to get access to any oxygen. And some of them are good at breaking down the complex carbs. And then a lot of them prey on the bacteria and will hunt the bacteria and eat them, so to speak. And their food, the host can eat some of the protozoa and other eukaryotic organisms. So bacteria have cell walls, protozoa do not. And so that's another issue with our digestion of microbes. It's tough to digest the cell walls. But if you have protozoa in you, there's no cell walls. There's anaerobic fungi that hang out. They're, eukaryotic organisms. And so they're doing fermentation in the rumen. And when they look at them, they got rid of their mitochondria because they live in such an anaerobic state that, and cytochromes are involved in respiration with oxygen. And so they're like, I don't need that. I don't have access to oxygen. So they got rid of the mitochondria and cytochromes. And so they're very much obligate fermenters with and totally dependent on their host. Okay, well, see, I'm gonna say a few more things about the rumen environment, but we ran out of time, so I'll save that for next time, and then we'll get into microbe-human interactions.